Welcome to Biology at Priyajit, my dear viewers. Today we explore a fundamental concept in cell biology, the extracellular matrix. Far from beginning just a passive scaffold, the ECM or extracellular matrix is a dynamic network of proteins and polysaccharides that supports cells, connects tissues, and actively regulates cell behavior. Its components play crucial roles in cell adhesion, communication, migration, and differentiation, helping tissues maintain structure and function. Let's briefly step into the vital framework that surrounds and guides every cell. So, ECM or extracellular matrix is the non-cellular material present outside the cell in all tissues and organs. We have already studied the internal structures of the cell. Now we'll study the extracellular material which provides a physical scaffold that supports and holds cells together. ECM regulates key cellular activities like cell growth, cellular migration which occurs during cancer, cellular differentiation which occurs during embryogenesis, the survival process of the cell, homeostasis or making the cell in equilibrium condition, morphogenesis, etc. And this ECM is composed mainly of water, proteins and polysaccharides. But ECM composition and organization vary between tissues and organs. ECM diversity, the variety of ECM develops through biochemical and mechanical interactions between the epithelial cells, fibroblast cells, adipocyte cells, and endothelial cells. This is the image showing the basic structure of the ECM. ECM is a highly dynamic structure as it continuously remodeled. It undergoes rapid enzymatic and non-enzymatic modifications. It also determines mechanical properties of tissues such as tensile strength, elasticity, and resistance to compression. ECM also maintains extracellular homeostasis and water retention. ECM in animals versus plants. In case of animals, it is a protein-rich ECM is there, and the main structural fibers include collagen and elastin, adhesive molecules like laminin and fibronectin. Plants, they have carbohydrate-rich ECM, and their main structural fiber is the cellulose, and the adhesive molecule pectin is also present there. Molecular composition of the ECM. ECM consists of three major macromolecular components like fibrous proteins as it provides strength, elasticity and structure. Collagen. We know that collagen is the most abundant protein in animals, approximately 30% of total protein, and it provides tensile strength. It supports cell adhesion, migration, chemotaxis, tissue development, etc. Elastin. It forms elastic fibers. It provides elastic recoil. It is found in lungs, skin, blood vessels, etc. Stretching and controlled by association with collagen. Fibronectin, it is very much important for cell attachment. It helps organize interstitial ECM. It influences cell behavior and tissue repair. Laminin is the major component of basement membrane. It promotes cell adhesion, migration and differentiation. The image showing the various ECM components, various proteins like elastin, collagen, laminin, fibronectin and proteoglycans like leucine or modular components, etc. Proteoglycans, it is also consists of core protein and covalently attached to glycosaminoglycans. It form a hydrated gel in the intercellular spaces. That means in between two adjacent cellular spaces. Its functions include water retention, shock absorption, binding growth factors, resistance to compressive forces. Major types of GAGs include hyaluronan, simplest GAG, it is not protein linked, chondroitin sulfate, dermatan sulfate, heparan sulfate, heparin, keratin sulfate, etc. Addition proteins link ECM components to each other and to various cells. Major addition proteins include laminin, fibronectin and pectin in plants. Laminin and fibronectin are glycoproteins. They mediate cell matrix interactions. Types of extracellular matrix. Basically, extracellular matrix types are interstitial matrix which surround cells in connective tissues, and it is rich in collagen, elastin, proteoglycans and fibronectin. Pericular matrix is found in the basement membrane. They are located close to cell surface. They are found in between epithelial tissue and connective tissue. They act as thin sheet-like anchoring layer, the protective barrier preventing tissue damage. Composition of the basement membrane include collagen type 4, laminins, nidogen 1 and nidogen 2, and proteoglycans include perlecan and agrin, and the collagen type is the type 15 and type 18. Basement membrane structure. Laminin network is found in cell adhesion. Collagen 4 network, they are responsible for structural stability. Both networks, that means laminin and collagen 4 network, are linked by connector molecules. So, what is the cell ECM interaction? Cells always interact with ECM via surface receptor proteins like integrins, discoidin domain receptors or DDRs, cell surface proteoglycans, and CD44, that is hyaluronan receptor. Functions of cell ECM signaling regulate cell shape, cellular movement, cellular function, and cell fate. Extracellular matrix. Proteases are the proteins which degrade proteins, so they basically break proteins by hydrolyzing peptide bonds. They are also known as proteinases or peptidases. We have already know about the matrix metalloproteinases, which are involved, which are overexpressed in cancer cells and help the break helps the cancer cells to break its surroundings and to migrate or invasive other tissues and organs. So human degradosome is approximately, there are 569 proteases according to Madoff's bioinformatics database. So based on the site of action, extracellular matrix proteases are of two types, that is exopeptidases which digest the protein from one end, either N-terminal end or C-terminal end, and endopeptidases act within the protein chain. They break the peptide bond within a protein. So based on catalytic group, metalloproteinases, they are zinc dependent, serine proteases, cysteine proteases, aspartic acid proteases, and threonine proteases. Role of ECM proteases, they are involved in ECM remodeling, tissue development, wound healing, and disease progression. Extracellular matrix receptors. Integrin are transmembrane receptors. They link ECM to actin cytoskeleton. They are important for cell adhesion, cellular migration, and signal transduction. Discoidin domain receptors or DDRs. This receptor is involved receptor tyrosine kinases. They are activated by collagen binding. They are involved in cell proliferation, differentiation, and tissue remodeling. CD44 or CD44, that is another transmembrane glycoprotein receptor, and it has no intrinsic enzymatic activity. So it is the main receptor for hyaluronan. It also binds fibronectin, collagen, osteopontin, growth factors, and MMPs. ECM targeting. Therapeutic importance of the ECM. ECM is altered in various diseases like cancer, atherosclerosis, autoimmune disorders, inflammatory diseases, etc. And the tissue microenvironment is very much crucial in this disease progression. Healthy ECM can suppress the tumor growth, and ECM disruption promotes also cancer initiation, progression, and drug resistance. Therapeutic strategies. ECM molecules act as drug targets. They improve drug diffusion and retention of water. They enhances treatment efficacy, reduces resistance, etc. Functions of extracellular matrix. Structural support. They provide strength and shape, prevents cell collapse, hydration and homeostasis. They can retain water, so they maintain the extracellular balance. They regulates tissue health, tissue shape, and help in the tissue morphogenesis by storing and releasing various growth factors. They protect and act as shock absorber, function as cushions to protect the mechanical stress. It protects the delicate cells also. Cell separation and organization, it maintains tissue architecture and keeps cells physically distinct. Growth and wound healing, which is essential for bone mineralization, supports scar tissue formation and helps repair damaged tissues. This is the image showing the various extracellular matrix protein functions. So thank you my dear students for joining today's class. As we conclude today, remember that the extracellular matrix is far more than a structural support system. Its components work in harmony to provide strength, elasticity and biochemical signals that guide cell behavior, communication, tissue organization. Understanding the ECM helps us appreciate how cells interact with the surrounding and how normal development occurs, tissue repair, disease progressions are regulated. So with this foundation, you are better prepared to grasp advanced concepts in cells and molecular biology. So keep revising, stay curious, and continue exploring the remarkable architecture beyond the cell. Thank you.